Hi everyone. Now I'm really happy to discuss how collaboratively we can build large neuroimaging data sets for glioma research. In this work corresponds to working group 2 of the GliMR cost action. As was discussed earlier, there are 50,000 new cases of gliomas per year in Europe. But if we look at the incidence rates locally, it is pretty low. This means that a lot of research studies who have looked into gliomas are unfortunately of limited sample sizes. They work with data sets that are pretty small. And in the following slides, I'm going to discuss why small data sets can be an issue for research in general by looking at different examples that do not come from brain imaging. But I will also look at how other aspects of how we build the data sets to build a model or to draw conclusions about a population. So my first example here was published, so it's a news article that was published in the Atlantic Science, and it's entitled "A Waste of 1,000 Research Papers." In here, the journalists discuss a study that was run recently that was trying to reproduce a landmark paper in genetics. That landmark paper was initially, the original one was published in 1996. And they tried to reproduce the results using a much larger data set. And by doing so, they couldn't reproduce the original results. And looking a bit more closely into it, one of the possible reasons was that basically the first study used a sample that was too small, and unfortunately the original results were a false positive. And this is a problem, of course, in science because we build on each other's work, and hence the title here of 1,000 research papers that were wasted. This has been discussed a lot in scientific communities under the notion of reproducibility crisis, and there is a growing interest into building larger data sets, of course in neuroimaging, but beyond that, in any field that is using data. So that's one aspect that is pushing us toward bigger data sets. But beyond small or big data sets, there are other aspects that we need to be very careful about. So here's another example from the New York Times, where they were reporting on a study that looked into facial recognition systems and basically identifies that these systems made a lot of errors when they were trying to recognize women or people of color. And looking more closely into it, they found out that basically the way these facial recognition systems were trained, they used data sets in which white male were predominant. And basically the model got really, really good at identifying people that look similar to the one in the training set, mainly white males, but not so good to identify women, for instance. And this is, of course, a big problem, and it reminds us that it's really important to have data sets that is representative of the population of our target population. Then a final example that comes from a running app. So a lot of people are doing running right now, and I'm sure a lot of you know, like they like running with their phone, and then they have these apps, and they get all sorts of metrics, including how fast they've been running, how far, and so on. And so the study here asked a very simple question. They compare how fast runners in their 20s were compared to runners in their 40s. But surprisingly, they found out that people in their 40s were faster than people in their 20s. And this is rather counterintuitive. So there was a lot of debate around it that I don't really want to go into. But one possible explanation that I found very interesting is the fact that if you looked at the people who were in their 40s and used the app in question, they were predominantly very good runners. So there were people who would run very regularly, who train for a marathon, and so on. On the other hand, people in their 20s who used the app were from a much more heterogeneous group of runners. You had very good runners, yes, but you also had beginners or people who would run for a month and then stop and so on. And basically, when you compared people in their 20s and people in their 40s using the data from this app, you would compare the two groups that were very different. On one hand, you had people in their 40s who were like super intense runner, and on the other hand, you had people in their 20s who went all the way from beginners to advanced level. And this is a typical example of selection bias, in which we find a result that is not true due to the fact that the data set itself had a bias, and the bias here was due to the fact that the data was collected through this running app. So this is another example of how careful we have to be when we build a data set to draw conclusions. So if we go back to Glioma Research, we've mentioned that currently many studies have unfortunately limited sample sizes. And the goal of Working Group 2 in the GliMR cost action is to try and build bigger samples that are going to be more representative of the diverse population. And the approach here is to look into data integration. So here's a quick illustration of what we mean by data integration. We often talk about data harmonization rather than integration. It's another approach where you are going to try and harmonize an acquisition protocol, the population, selection criteria, and so on. You could build a mega study across Europe trying and doing that. On the other end, data integration is more about reusing the data that is already there, working with many small studies and combining them together to build a bigger study. So let me try and give a little bit overview of what we mean by data integration. So here first, we start with the outline of how our our small sample studies is going to work. So we have some raw data, have a feature extraction stage, or sometimes called pre-processing. We get some derived data we think are interesting to work with. We do a statistical analysis or machine learning analysis if you're into it, and we get some results. So you have a study that is extremely homogeneous. The data is going to be acquired at a single site and analyzed in a single lab. So that's one extreme. Now, one way we can start doing some data integration is to find some raw data that is similar to mine and just inject it at the level of feature extraction. So let's say I'm based in Rennes, I'm going to have a collaborator in Edinburgh use their data and inject it in my pipeline. Of course, here we're going to have more variations. The two types of raw data are going to be different. They've been acquired on a different machine. Maybe the ESMRMB sequence were slightly different, and so on. We could also work directly with processed data. So that could be the case if raw data cannot be shared, for instance, for privacy reasons. That could be the case because it's just more computationally efficient to reuse data that was already, where feature extraction was already done, and so on. And if we do that, again, we have to take into account other level of variations coming from the fact that the feature extraction stage was done somewhere else, maybe using a different tool on a different platform, and so on. Another example is meta-analysis that has been going on for longer in the field, where you combine results from different studies into a new study. When you do meta-analysis, your different studies have used different methods, data that come from different sites, and so on. So you have a lot of variability to take into account. So here's the approach. The idea is that we're going to be able to reuse data that come from different sites, and that were required to do maybe different studies, but this comes at the cost of more variation that we will have to find a way to take into account. So now I'm going to get more specifically into GliMR Working Group 2, which is trying to enable multi-site data integration. This working group is led by myself and Cyril Pernet, who is based at the University of Edinburgh. And we are focusing on three main directions: data privacy, data infrastructure, and data portability. And I'm going to talk a little bit more in details for each of these. So data privacy first. The goal is to find a way to inform participants and collect their consent for data sharing in compliance with GDPR, because of course in Europe we have to comply with GDPR. So within GliMR, our goal was to review existing standards on data privacy. So far we've been building on an existing project that has been going on for a few years now, which is Open Brain Consent. Open Brain Consent was initially initiated by Yari Halchenko, and the goal was to collect sample consent forms that came from different labs that had been approved, had been used, and enabled data sharing, so that other researchers who would try to share their data could just look into them and maybe use some of the text for their consent form. And more recently, last year, Stefan Yunis adapted this work, which is like, it's a worldwide collaboration between researchers, anyone can participate. And Stefan was looking more closely into how we can adapt this consent form to be compliant with GDPR so that they can be used in Europe as well. And a lot of work has been going on into this. There was a workshop organized with GliMR, and I'm happy to share that there is now a preprint which is pointing to sample consent forms that enable data sharing and work with GDPR. There are currently, I think, 11 translations, so you have the languages here. If your language is not there, we'd be more than happy for you to join the effort. And so this is one of the things we do with this working group too. Another direction is data infrastructure. So more practically, which tool can you use, which database are available for me to go and share my data. So the idea is to review existing data sharing infrastructure with a particular focus on Europe. And here we are collaborating closely. We've been so far collaborating closely with NBIT, the European Network for Brain Imaging of Tumors. That was an initiative started by Daniele Marinazzo and Cyril Pernet. So they do have a database behind that website that let people share their data, and this is based in Europe. But of course, we'd be happy to hear about, to work with other efforts to make a review of all these platforms that are available for researchers. Then the third point is data portability. So we've touched upon data integration and how we're going to have to deal with the fact that our data are much more heterogeneous. But before we can build methods that let us work with this heterogeneity, we need a way to convey this information. I need to be able to know these data was acquired on this particular scanner in that location with this sequence, and then pre-processed using that tool and this particular algorithm and so on. And so to do that work, which is to be done, we're going to extend the international BEAD standards. So BEAD stands for Brain Imaging Data Structure. It's a very nice community with more than 200 members right now. So I've only put the picture of the current steering committee. Initially, BEAD started with MR, they've then extended to EEG, MEG. They're working into PET. So there is it's really a community that is very active and a nice place to try and see how we can build standards for advanced MR biomarkers. So I've given an overview of what we are doing in GliMR Working Group 2. If you have an interest in any of these, we'd be very happy to hear from you. So our email address is here. You can join the working group and you'll get all the information about the events that are going to happen. If you want to be connected more specifically with one of the projects that I've mentioned, we'd be happy to do that as well. And I'd be happy to answer any questions, of course. Thank you.